Hello, you are listening to 1 in 2, a Manchester Cancer Research podcast, brought to you by the University of Manchester and the Manchester Cancer Research Centre. With 1 in 2 of us receiving a cancer diagnosis at some point in our lifetime, it has never been more important for our research to improve the outcomes for people affected by cancer. I am your host, Sally Best, and throughout this series I will be speaking with Manchester cancer researchers about their innovations, discoveries and projects that are changing the landscape of early detection. In this episode, we speak to Professor Sarah Cartmell on her work in bioengineering and ex-vivo modelling, which involves studying cancer cells outside of an organism. Specifically, we talk about her team's work developing these models that can analyse in 3 dimensions how non-cancerous lung cancer cells develop into tumours and what the findings from this work could mean for improving patient outcomes. Hello, Sarah Cartmell. Thank you so much for joining me. How have you been?
I'm good, thanks. Yeah, it's a pleasure to be here.
Pleasure to have you here. Absolutely loving your presence. Sarah's in a lovely dress today. I'm very jealous. I am, of course, dressed in black because what else for this Manchester weather? But, you know. So I'm wondering to start off with, if you'd be able to tell me and the audience a little bit about yourself and what it is that you do.
Yeah, sure. So my name, as you just heard, is Sarah Cartmell. I'm a Professor of Bioengineering at the University of Manchester. I'm also the head of the Department of Materials and the Faculty of Science and Engineering here at the university. My labs are located in the National Royce Institute of Materials, also based here in Manchester.
Okay, and for the benefits of the audience, we can put a map on because the Manchester campus is very broad and has a lot of different buildings, so it'll be helpful for you guys to look at that. But I mean, I'm intrigued to ask, what does your day-to-day look like?
So typically it's a lot of emails, a lot of meetings. I do have quite a large research group. So I've got about 20 researchers, PhDs, postdocs, project managers within my group. So they're normally in the lab going through, collecting the data, and we sit down and go through the data. So there's a lot of exciting kind of project discovery and sitting down and coming up with new hypotheses based on the data that we've gathered.
Just to kind of go back, you're a professor in bioengineering. We've had quite a few different people on here that aren't your kind of standard cancer researcher, but I'm wondering, what's your research journey into engineering?
Sure. So I began my academic career at the University of Liverpool in 1993. I studied clinical engineering with material science there, and that was a Bachelor of Engineering degree, but that was perfect marriage for me. So that combined my engineering passion as well as my passion for medicine. I carried on at Liverpool to do a PhD. And I studied the degradable phosphate-based glass and its use for urinary stress incontinence and tissue engineering purposes. 2000, I moved to Georgia Tech in Atlanta in the States for a couple of years, and I was in the mechanical engineering school there. So I keep moving from different faculties, very interdisciplinary work. Here, I really began my interest in orthopaedic tissue engineering, which is kind of these 3D tissue models that we'll be talking about today. I moved back to the UK in 2002 when I started working at Keele University for about 8 years, and then I moved to Manchester in 2010.
Can I just say, Sarah does not look like she's lived this much life. 2002, I think I was 4. Goodness me, it's a lot. And I mean, it's so interesting to hear about. You work as the head of the Department of Materials at the University of Manchester, is that right?
That's right.
Okay, your bio is right online, love that. And I'm wondering, within this role, how many different fields do you work across? So you've kind of mentioned healthcare and medicine and things, but like, what's the kind of plethora of roles?
So I have obviously a couple of different roles. One is the head of the materials department. The materials department itself is very broad in its discipline. So Manchester is a cotton trading town. It initially came from technical textiles. So the university in 1824, nearly 200 years we've been going, is built up from that technical textiles, and we carry that on in our department. So we actually have material science, as you would imagine it, in terms of, as I mentioned, textile technology, corrosion, composites, biomaterials, which is my field, imaging and characterization. But also we have a large body of research and teaching in fashion business technology. So there's a lot of kind of humanities, business, and obviously more arts within the department as well. Myself, in terms of my research, I work in biomaterials. So I work across the Faculty of Biology, Medicine and Health and the Faculty of Science and Engineering, and sometimes humanities as well, because what we're doing affects society, as you can imagine.
It's a massive plethora of work, and I really want to focus on cancer early detection because that's what this podcast is about. But I mean, just to get a full idea of what you spend your time doing, do you have any examples of a project outside of cancer that you work on that you'd like to kind of briefly highlight, just to give us a little tidbit into the life of Sarah Cartmell?
Sure. So last year I started my own spin-off company called Retendon Limited. That's been translating some of the tendon repair products that I've been developing in my laboratory over the last 10 years. We've got a couple of products that are made from a degradable plastic called polycaprolactone. And one of these products is to augment tendon suture repair and therefore improve the functionality of the wound healing and the time needed for the repair to shorten that. And the other product is a novel tendon retrieval and attachment device. I've also got a portfolio of research in wound care products, and I also investigate the effects of applying electrical regimes and mechanical forces to cells and tissues in order to influence their activity. So we can either get them to grow more, to proliferate, or to differentiate, which means turns into different cell types, or produce more external matrix, making more tissue, if you like.
God, what don't you do? Tendon repair. That's insane. I love that. I need to talk to you more about that another time. I'd like to kind of get to the nitty gritty, but I mean, how long have you been here in Manchester? So since, when did you say you moved over? 2002?
2010, University of Manchester. But I'm from Manchester myself. I'm from Sale, from South Manchester.
Oh,
so,
nice. I'm from Stockport.
Oh, fantastic.
two different ends of the woods, but Manchester through and through. We love it. So you came back.
I did, yeah.
You came back. So you've been here, what year is it now? 2022? Mm-hmm. I accidentally wrote 2020 earlier, but 2022. But yeah, 2022. So that's 12 years. And I mean, compared to other places you worked, what is it like to work across this kind of broad research landscape that is Manchester? Because I mean, it's massive. It's, you know, there's so many different counterparts of the Manchester campus, and I'm just wondering, what's it like?
Yeah, it's really exciting. So the depth and the breadth of the research activity that we have at Manchester is one of the reasons why I came here to work. The opportunities are really vast, and the expertise and the resources that are all here on our doorstep. As such, we're in a really great position to translate our research through its inception all the way through to the clinic. And
do you find it easy to collaborate here because there's so many different teams? Like, has that made your research easier?
It definitely has. If anything, there's probably, because I get very enthusiastic about different projects, probably too easy to collaborate. I have to make sure that I'm not taking on too much and overcommit. That's the only thing.
Yeah, I understand that. I get very excitable, but, sometimes it's the way of the world. And I mean, coming back to what I'd like to talk to you about today, I'd firstly like to ask, when and how did you get into working within cancer early detection?
Yeah, so there's a guy called Rob Bristow, and he joined the University of Manchester in 2017 as the director of Manchester Cancer Research Centre. So he began to bring together different expertise across the university, such as biomaterials, that could be a benefit to the cancer research field. And he held a variety of different workshops. Some of those I went to in order to mix with different cancer research academics I hadn't previously collaborated with. I then began translating the 3D tissue engineering models that I've been developing all through my career for cancer early detection models when we secured our first grant in 2019. And that gave us the resource to fund an excellent PhD student who's with us now called Emmanouela Mitta. Big shout out for Emmanouela. She's working with myself and Andrew Gilmore and Angeliki Malliri, who are at Manchester University as well in the Cancer Research Centre. And Emmanouela is looking to create a lung cancer early detection model. Since that project, so that's ongoing, I've also been working with a lung oncologist, an academic clinician, Sean Knight, and cancer biologist Katie Finnegan, who were also working on lung cancer. And we've just been awarded a couple of research grants that we're just starting now.
This is fab. I love it. Loads of people. So, I mean, just really quickly, biomaterial, that's a biological material, just for the kind of sake of me and the audiences.
Yes, just to clarify, so when I say biomaterial, I mean a material that's going to be in contact with a cell. So it doesn't necessarily mean, sometimes you can read in the literature, biomaterial is a naturally occurring material. When I say biomaterial, I talk about, it could be a naturally occurring material, such as collagen, or it could be a synthetic material. It could be something that we've made in the lab from different chemicals, but the purpose is biological.
Okay. And in terms of, say, just back to your tendon work, it would have to be a material that wouldn't negatively interact with the cells in your body.
Typically, yes. So normally we want it to be biocompatible and non-cytotoxic. It depends on its application. You might want a biomaterial to create an adverse effect, say if you wanted it to kill cancer cells, for example.
Okay, interesting. And I mean, back to this kind of modelling, how do you do it? So you've said that Emmanouela, she develops these lung cancer models, and I know you're going to kind of go on to this, but I mean, how is that done? Is that done in the lab? What do they look like?
Sure. So we do have a lot of different rooms in our laboratories for all the different purposes. So one of them is like a wet chemistry lab where we're able to manufacture the different materials that we want to use. We call the materials that we're developing scaffolds, and we create those materials, as I mentioned, so they're typically for this purpose they're biocompatible, but also they've got the similar shape, the size, the chemistry, the mechanical properties that we need for the particular tissue type that's in our body. We then take healthy cells, so non-cancerous cells at that point, and grow those. We place them, we call them seeding, put those onto the scaffolds. And what we tend to do in the cancer early detection is in half of those samples, we're able to keep those as controls, if you like, and keep those healthy and non-cancerous. And in the other half, we're able to we're able to create like a cancerous tumour. So we're able to genetically alter those cells using a process called transfection. So we can create cancerous tumour. And as such, we're able then to analyse those very early changes in cancerous versus non-cancerous cells, but in a 3D environment, in a developing tissue, rather than just on the bottom of a plastic dish, which is essentially the process that's happening now. So it's a lot more kind of biomimicry. It's mimicking the biological environment that we have right now. The materials that we use hugely vary depending on what environment you want to create. So it might be a gel that we want to encapsulate the cells fully, or it might be a fibrous mat where we place the cells directly on top of them. And then we can then apply different types of stimulus to the samples, such as mechanical forces to emulate a breathing action, for example.
And can I just ask, where are these healthy and cancerous cells? Where do you get them from?
So again, it depends on your research question. So we have, you can use something called a primary cell, which is where we take a biopsy of cells, of tissue, and then we isolate those cells, we digest them out. And typically, I'm very interested in that clinical translation. So typically we would use biopsy. Obviously, we have ethics in place, ethical forms and approval in place, typically human samples to make that clinically relevant. A research question might be comparing some of the data to literature that's already out there, and that might be from a mouse model, for example. So it might be that we need to take a skin sample or a blood sample from a mouse, or you might be using a cell line, which is something that's already established but had been established many years ago by a different researcher.
Okay. And I mean, so are these what we'd call ex-vivo models?
So the terminology of ex-vivo and in vitro does change from field to field, which is something I've had to be aware of when I've started very much with this interdisciplinary working. When I do talk to the cancer biologists, then when they say ex vivo, they're talking about something that I mean in vitro. So for me, ex vivo means when you've taken a piece of tissue, maybe a piece of skin, and we work with that piece of tissue in the lab. So we haven't isolated any cells from that, but it's still an intact piece of tissue, if you like. It's ex vivo, it's out of body. So, and we can then perfuse that with blood and keep it viable for another 24 hours and keep that going. So that's an experiment in itself. The work that I do tends to be with 3D in vitro models, which is when we use single cells, we seed those onto those 3D scaffolds that I mentioned earlier, and then we build up the 3D tissue. So we create that 3D tissue by growing them in containers that we call bioreactors. And those bioreactors then control the environment that's needed to grow that tissue, essentially the different gases, nitrogen, oxygen, carbon dioxide, all the different nutrients, the salts and the sugars that we need for those tissues to develop, different proteins and different, stimuli such as mechanical forces or electrical regimes that the tissues need to grow. I have an example. So Emmanouela, that I mentioned earlier, Emmanouela Mitta, who's our PhD student, she's working with a type of scaffold which is made from a biocompatible rubber. It's called polyurethane. And she's created these different fibrous mats out of this polyurethane using a technique called electrospinning. So they're very, very fine, kind of randomly aligned fibres. And she's then taken those mats and grown lung epithelial cells on those. Okay. And so she's either taken these healthy cells or genetically manipulated cells, as I mentioned earlier, and she's growing that in a bioreactor system. So the cells are in an air-liquid interface. Obviously, with your lung, you have, obviously, there's fluid there, but there's also air. There's also a gaseous presence. So Emmanouela's currently at the stage where we're now incorporating magnetic particles into these polyurethane fibres. And by doing this, we can then apply an external magnet and then move the rubber mat, if you like, and stretch it and apply a physical strain to the cells that are seeded onto the fibres. In a way, then it mimics the motion of a lung alveolar. So it mimics that breathing action. And in this way, we're able to analyse the samples that as the cancer tissue and the healthy tissue grow in comparison, to each other in this lung alveolar mimic in the lab.
Fab. And I mean, just for the audience that don't necessarily know lung physiology, could you just explain briefly what an epithelial cell is and what an alveolar or alveoli are?
Sure. So if you like, I guess if you look at a tree where you have the main trunk, and that might be your main trachea, your airway, and then it goes off and it branches off, and those branches get smaller and smaller and smaller, and kind of at the end of those twigs, if you like, there's this little circular sac, and obviously it's like a bag. When you breathe in, it's expanding, and when you're breathing out, it decompresses, and so you've got your negative and positive pressure going on in your lungs. The alveoli are these tiny little sacs right at the end of those branches of the tree, if you like. So they're very small individual spaces, but they're very important in terms of increasing the surface area of your lung. And as cancer develops, if that gets blocked, that obviously has an impact with the exchange of the oxygen. You asked me about epithelial cells. So within your skin, within your lungs, you have different types of cells, and epithelial cells is one of those cells which is on the surface, layering on the inside of your lungs. And obviously, would be if you had lung cancer, sometimes those epithelial cells, those surface cells on the inside of the lung, will be contributing to the tumour growth.
Okay, fab. So what I've heard so far is that you're doing this kind of modelling type. And one is having your control group, which is looking at non-cancerous cells, and the other is having your non-control group, which is looking at cancerous cells. How is that being applied in early detection? Are you looking at different cancer markers at different stages of development? Like, what's the process behind that?
So what we're trying to do is, as you can see, we're able to then develop the cancer from a very early stage all the way through. If we can detect distinctive biomarkers that are emitted, if you like, by those cells. So the cells will have a different genetic profile, different proteins that they're producing as well. And our hypothesis is that there's a signature, if you like. So it's not just, I don't think it will be just one it might be, but I don't think it will be just one protein that we can then say, oh, this protein's been expressed, therefore the cancer has developed. I think it will be a signature. So I think it might be 12 different proteins, for example, that would be upregulated, that would go, oh, that's unusual. That wouldn't happen in a healthy tissue. With the hypothesis then, if we have that signature profile, we could then possibly take, for example, a blood sample from somebody who's more at risk. So that might be somebody who's genetically predisposed to getting particular lung cancer, or it might be somebody who's smoking heavily, and that might need regular checks to see if that signature, if that starts going up, then that might, again, that's the hypothesis and what we're trying to do.
Okay, so you're saying that you're looking out for these biomarkers, which I'm assuming are cancer markers as a kind of lay term.
That's right.
And then they might be things such as proteins that are upregulated, i.e. kind of overproduced, and they signature the start of a cancer that you wouldn't then see in a healthy patient with non-cancerous cells.
Exactly.
Okay. I mean, that's amazing. And I mean, it's great to see like why we're using these sorts of models in early detection, and that also clinical application of blood sampling and being able to test at early stages and look at people that are at risk, because I think that's a major thing in terms of risk factors. And I know they're kind of catching it at that stage one, which is where you're saying would be these kind of starting upregulated upregulating of these biomarkers is just amazing, because I guess it kind of means that your treatment options are more kind of better and more varied and less kind of invasive as well.
Absolutely. And the outcome, obviously the success then of that treatment would be, hugely increased.
Okay. I mean, it's great. And just taking a step back here, this is kind of a biological question, but I'm wondering if you'd be able to explain how a cell goes from being non-cancerous to cancerous.
Yeah, so our bodies are made up of trillions of different cells. They're grouped, obviously, as I mentioned before, into different tissues and different organs. And there are genes inside the nucleus of each of those cells. And those genes tell the cells when to grow, when to work, when to divide, and when to die. And normally, our cells follow these instructions and we stay healthy. But when there's a change in our DNA or damage to it, a gene can mutate. And mutated genes, they don't work properly because the instructions in their DNA gets mixed up. This can cause the cells that should be resting to divide and grow out of control, which can then lead to cancer. When the genes aren't working properly, they tell the cells when it's the right time to grow and divide. And when the cells divide, they make exact copies of themselves. So one cell divides into 2 identical cells, 2 goes into 4, and so on. In adults, the cells normally grow and divide to make more cells only when the body needs them, such as to replace aging or damaged cells. So there's not a huge, apart from stem cells, there's not a huge amount of division that's going on. But cancer cells are different. So cancer cells have gene mutations in them that turn the normal cell into a cancer cell. These gene mutations they might be inherited, they might develop over time as we get older, excuse me, and the genes wear out, or they might develop if we're around something that damages genes, such as cigarette smoke or alcohol or UV light. A cancer cell doesn't act like a normal cell, as we discussed earlier. It starts to grow and divide, that gets out of control, and instead of dying in the way it should. They also don't mature as much as normal cells, so they stay immature. And although there are many different types of cancer, they all start because the cells are growing abnormally and out of control. And cancer can start in any cell in the body.
Okay, and I mean, I'm guessing in terms of the difference between these cancerous and non-cancerous cells in your models for early detection, that's kind of the start point. Are you using lots of different kind of cancerous cells? Like, are they from different places? Do you witness different levels of growth and mutations in different cells from different areas?
So there's lots of different researchers working in this area. So I myself have only just, as I say, started working in the lung field, and we're doing that by the transfection and then causing that cancer. So I do work with other researchers that have, as you've described, have all these different stages and different types of cells that are cancerous and grow them in that way. But again, what I'm doing is initiating that transfection and starting that cancer in the lab, and that's what we're observing.
God, it's so cool. Do you like watching it?
It depends what you mean by watching it. I like looking at the data. I think if I just stood looking down the microscope at the cells, that's a bit like watching paint dry. You'd have to watch, a speeded up movie.
Yeah.
And then that's pretty cool, yeah.
How much would you have to speed it up by?
Well, that's a good question. So the cells, a typical cell might double, in 24 hours. Okay. Cancerous cells, again, if it's a younger patient, that can be quicker or it could be longer. So if you take it like a healthy cell, for example, if you took a bone marrow stroma cell from a 19-year-old male, then you'd be looking at 24 hour turnaround in terms of doubling of cell number. If you took that from a postmenopausal female, for example, you're probably looking at 72 hours.
Goodness, so cool. Get me in the lab looking down these microscopes. I love a microscope. And I mean, so you've mentioned that you've started to develop these models in lung cancer. Have you developed these in any other cancers? Are you hoping to develop them in any other cancers?
Well, that's the plan. I think what we have is a plethora of different materials and 3D bioreactors that I mentioned, those environments, that really we can apply to a whole variety of different tissues. So we're talking now with our researchers in the Manchester Cancer Research Centre and also through AKID, the Alliance for the Cancer Early Detection, to talk about what we can do. What do they need that hydrogel that I mentioned before? Do they need that 3D environment? Do they need to control hypoxia, which is essentially how much oxygen is getting to the cells, and control that permeability, because that's important, obviously, as tumour develops. Do they need to alter the mechanical environment? So you might get a breast tissue, for example, that gets more fibrous and gets very stiff. And a lot of the models that are used right now wouldn't counter for that change in mechanics, but that could be key.
Okay, so there's a lot of kind of trial and error behind it. That's really interesting. So aside from the tissue sampling that's being used, do these models vary between, say, the work taking place in lung cancer or breast cancer?
So they would change quite a bit, yes, because the different tissue, obviously the different cell types are going to be different, and the way the cancer develops in terms of the tumour is going to be different, and the mechanics that I mentioned is different. So we would probably use a different scaffold, if that makes sense. So I know that one of my colleagues at the University of Manchester, Alberto Ciani, they're developing this kind of peptide-based hydrogel, and they're encapsulating some of those breast cancer cells in there and seeing how that develops as well.
Okay, and just to confirm, peptide is basically protein.
That's right. It's like the building block of a protein.
Building block of a protein. Can we have biology lessons with Sarah Cartmell, please? That should be a new podcast series. And I mean, you've mentioned ACE, which is International Alliance for Cancer Early Detection. And I know quite a few of the people on here have done work with ACE. Great group. But I'd like to know, number one, so is the project that you've specifically been working on with ACE this development of the lung cancer model?
That's what I've been doing so far, yes.
Okay. And I mean, how did the partnership form?
So again, it was led. The initial collaboration and introductions, Rob Bristow, again, is key. So he knows a lot of the people that obviously that I wouldn't know, and is putting us in contact. We also have within Manchester, it's kind of a network, this Advanced Materials in Medicine network, and we also have the Royce Institute that I mentioned before. So that's already linking us with different materials expertise that we might not already have in the team, but we know that we need. We already have connections with the University College London, for example. So we've been working with Emmanouela and one of the lung cancer projects that's just about to start that I mentioned with Katie Finnegan and Sean Knight. But essentially, they start with, getting people together and presenting the research that, right, this is the unmet clinical need, this is where we are, this is the state of the art, this is what's going to be big science, this would what gives a breakthrough. And then we can go off and sit down and go, well, we could approach it through this, particular technique, and whether or not that is a new 3D model that I mentioned before, an inclusion of mechanics, or it could be, in our faculty, we have lots of really cool new analytical equipment like microscopes or spectroscopy that are cutting edge that wouldn't normally get to other fields maybe for 15 years, right? Because they're so state of the art. That wouldn't be a common tool in the lab, if you like. So that's how we can, through those conversations, we can find out that research need, get to this specific research question, and then come up with some solutions.
So, I mean, within the ACE project, do you work with partners in the US on this project? Is it just partners in this country?
Right now, we're only working with Emmanouela at UCL, but it doesn't mean that's because we don't want to. We just, as I say, I've only just started a couple of years ago in the field, getting some data. We're going to be presenting those at conferences soon. So Emmanouela, who's my PhD student, I mentioned, is going over to Portland, Oregon in a couple of months, going to present the data. We're hoping some new collaborations are going to come from that as well.
And in terms of the unmet clinical need for this project, was that just late lung cancer diagnosis?
Again, it's going to who initially the collaborations that were formed. So the researchers that I've been working with, Andrew Gilmore and Angeliki, really are saying this is what's needed, this is where we are right now, and this is what would make a big difference. So yes, they were working in lung cancer. Obviously, Andrew works in breast cancer as well, but this particular model was working in lung. But the same would be the same for any tissue.
Yeah. And I guess in Manchester, there is that massive unmet clinical need just because of the vast rates of lung cancer in this population.
That's right. It's a huge, huge demographic, yes.
So there is kind of that massive need to address that, and the work that you're doing is obviously really contributing towards that. What do you hope that the development of models such as these will mean for patient outcomes? So
obviously we want to improve the patient outcomes. We want to be able to have a fast and easy, a reproducible way of detecting these cancers as early as possible, that would then mean that we have better treatments, better outcomes. We want to really get to a place where we can move to a world where cancer isn't a scary word, you know.
Yeah. I think that's the thing, and like this is this podcast name 1 and 2, and it's, yes, 1 in 2 people will be diagnosed with cancer, but I think it's something like 1 in 2 die from cancer that are diagnosed, and it's those rates that we're really looking to improve. So even if it is the 1 in 2 that are still diagnosed in 20 years, 30, 40, 50 years time, it shouldn't be seen as the same death sentence that it has been in the past, because it's all about the early detection side of things and the improved prognosis. And it's just amazing kind of speaking to people about the work that they're doing, because it is removing that death sentence from the word and making it a less scary cancer, you know?
Absolutely.
So, I mean, Sarah, what makes you passionate about your research, and specifically cancer research?
Well, obviously, just what you've just said, right? The impact. Obviously, I love doing what I do. I find it really interesting, but actually having that impact at the end really is what motivates me. I love coming up with solutions. So people coming to me and say, this is the problem that I've got. You know, what can we do? And then go, right, well, we could solve it this way, this way, this way, and then coming together as a team. So again, I really like working with people from other disciplines. So clinicians, industry, mathematicians, biologists, you know, material scientists, engineers, all sorts. That is such a stimulating environment. Everyone's coming from a different viewpoint, and I'm learning so much every day as well as contributing, right? So it's very exciting.
Yeah, I guess that's the thing, having external people coming in. You do think in a different way, and you're forced to, but you also force them to think in a different way. And it's just, yeah, it's amazing. I love the research kind of landscape here in Manchester, because it is just so vast. There's so many different groups, and it's so easy to collaborate here, because there's just so much going on. And like, there's a lot of like-minded people like you who have that drive for change and, yeah, wanting to kind of make a difference. Have you got anything in the woodwork? Anything coming up? Any new projects? Doesn't have to be cancer related.
I think the things we're translating right now, obviously, is the tendon, which is quite new with a new spin-off company. I briefly mentioned some of the work I was doing on electrical stimulation on cells. So this is really exciting in that, you know, our body has electrical regimes associated with it, and most people will be familiar, you know, with the heart, maybe taking an ECG, or the nerves with those electrical synapses. But each of our tissues and our cells has a electrical regime associated with it. So what we're doing in my lab is applying specifically targeted electrical regimes to stem cells, so mesenchymal stem cells that we take from people's bone marrow. And we're able to apply the right regime and turn them into different tissue types. So we're able to turn it into bone, or we're able to push it towards chondrogenesis and things like that instead, which is really exciting.
Fab. And just to confirm, stem cells are the kind of the first cell that you'd have in terms of a divisional pattern of a cell. So
That's
it's
right.
what you start with.
That's right.
It's the flower to the cake.
They're the kind of the baby cells that grow up and mature into, you know, your muscle, your fat, your bone.
Yeah. And they can be anything they want to be, but they are obviously directed towards certain specific cells. My analogies are getting out of hand. I mean, I ask this to everybody, but I think it's a really important question. And like, looking to the future, what are your hopes for this modelling? And yeah, how do you see it progressing in your dream world?
So, as I mentioned before, we're very interdisciplinary. I just really hope that all the biologists and the engineers really continue to work together to grow in our understanding of each other's fields and create even more synergy. We've really got such potential to create relevant testing models in the lab. We also have increasing expertise and ability, as I mentioned before, to analyse the samples from an engineering perspective. And in these ways, I really hope that we improve our understanding, but also increase the rate of gaining data, and therefore the solutions faster, so we can again move to that world that I mentioned before. That's cancerous and that scary world.
Yeah. Oh, great stuff. I mean, thank you so much for taking the time out of your day to come and speak to me. It's honestly just great, so appreciated, and I learn a lot from this, as well as hopefully the listeners. But I mean, yeah, as usual, I'll link Sarah's work in the blurb, and kind of put on a few diagrams as well, just so you can see that whole cancer research landscape. But yeah, thank you so much.
Thank you for having me, Sally. Thank you.
If you have been affected by anything you've heard in this episode, please see the show notes for a list of charities and organisations that can help.
1 in 2 was brought to you by the University of Manchester and the Manchester Cancer Research Centre. Listen to our next episode to hear from more of our researchers as they share the innovations, discoveries and projects that are changing the landscape of cancer prevention, early detection and treatment. To find out more about what you've heard today, please see the show notes for this episode, where you'll find a transcript and links to further information and research. Cancer is one of the university's 5 research beacons, showcasing the interdisciplinary collaborations and cross-sector partnerships that are tackling some of the biggest questions facing the planet. To hear more about Manchester's research in advanced materials, biotechnology, cancer, energy and global inequalities, go to manchester.ac.uk/beacons.